Good day everyone, we are the Group 3 and we will present the concept of Proteins Part 2 plus Enzymes. Disclaimer: the presenters do not own any concept and image used in this lesson. The concepts and images involved in this presentation came from various educational resources which will be solely used for educational purposes. What is Bradford assay? The Bradford protein assay is used to measure the concentration of total protein in a sample. The principle of this assay is that the binding of protein molecules to comacidi under acidic conditions result in a color change from brown to blue. What is Bradford assay? Bradford assay is a colorimetric assay. It depends on the color of the solution. It uses spectrophotometer, and it's a quantitative assay to measure the protein concentration in a solution. The principle of Bradford assay. Bradford assay depends mainly on this molecule, which is called the Kumasi brilliant blue. Kumasi brilliant blue is a very special molecule, and it has a character which is used for the Bradford assay. Kumasi brilliant blue can exist in two states. The first is the unstable state, which is the cationic. The second is the stable state, which is the anionic. The very special about Kumasi brilliant blue is that the two states don't have the same color. The unstable state is reddish to brownish, like the color you're seeing here, and the stable state is bluish like the color you're seeing here. So, this is the character we use for the Bradford assay. Now, how does it work? Okay, so in here we have a solution of proteins, and for example, you want to know the concentration of your proteins in the solution. The Bradford assay will give you the whole concentration of your proteins in the solution. So we have a protein solution here. Now we will take a sample of this protein solution and mix it with the Kumasi brilliant blue in its unstable state. The medium is acidic. The proteins mainly are positively charged in an acidic medium. Now, the Kumasi brilliant blue has a lone electron pair, and this lone electron pair, it can give this to the protein. And where exactly to the ionizable group? The protein is a chain of amino acid, and each amino acid has a side chain. The protein can have or may have many ionizable groups in its amino acid. The Kumasi brilliant blue can give the protein this lone pair, and in this case, when the Kumasi brilliant blue give the protein a lone pair, the protein becomes unstable. And then we have in the solution an unstable Kumasi brilliant blue and unstable protein. Now the Kumasi brilliant blue is mainly negatively charged, and the unstable protein is mainly positively charged. And then these two unstable molecules, they need to bind to each other in order to be stable. So they want each other first by ionic forces. And then the attachment will be further worsened by a hydrophobic van der Waals forces. Why? Because the Kumasi brilliant blue has a nonpolar region, and the unstable protein has a hydrophobic pockets on its tertiary structure. So they can also bind to each other by van der Waals forces. Now this binding, let's say between the protein and the Kumasi brilliant blue, makes them to be stable. And when the Kumasi brilliant blue is stable, it's not reddish anymore, but it turns to be bluish. And this is the main principle of the Bradford assay. This blue color depends on the amount of proteins in the inside of the solution. So when we have a little bit of proteins, the bluish color will be light. And when we have more proteins in the solution, the blue color will be darker. So the darkest the blue color is, the more protein we have in the solution. The materials for the measurement of the protein concentration are bovine serum albumin, Comasi brilliant blue, methanol, and phosphoric acid. Now, how can we measure the concentration of the protein in the solution? We need a calibration curve. And how to make the calibration curve? We take another protein of a non-concentration, mainly we use BSA. So BSA is a protein used to make the calibration curve or the standard curve. So to make the standard curve or the calibration curve, we use BSA, bovine serum albumin. We take a BSA solution of a known concentration. Let's say we have like 2 nanogram per microliter, and we produce several concentration or several dilutions of this Comasi brilliant blue. What we get is this. Let's say this is a Comasi brilliant blue without any proteins here. Let's say we add 1 microliter of this solution. We add 2 microliter, 3 microliter, 4 microliter. And then we will have like serial dilution of the BSA protein, like 2 nanogram per milliliter, 4, 6, 8. We can produce more and we can make it as much as we want. Let's say the first one is a Comasa brilliant blue without any proteins. Let's say we add 1 microliter of this solution. We add 2 microliter, 3 microliter, 4 microliter, and then we will have like serial dilution of the BSA protein, like 2 nanogram per milliliter, 4, 6, and 8. And we can produce more, and we can make it as much as we want. If you look at the color here, when the Comasa brilliant blue is alone, it's like reddish. 2 nanogram is not so obvious. The bluish color is not so obvious. Then 4 nanogram, it becomes more and more bluish. Now we take this and we measure the absorption of this solution in a spectrophotometer on 595 nanometer because we want to see how much bluish color we have in the solutions. We use the Comasa brilliant blue as a blank in the spectrophotometer, and then we measure the absorption of the other solutions. Then we will get something like a curve. So in the y-axis, we have the absorption of the solutions. And in the x-axis, we have the concentration. As I said earlier, we can have as much concentration as we want. Normally, the absorption should not be more than 0.1. If you get an absorption more than 0.1, then you should dilute your protein sample more. Because when the absorption is higher than 0.1, it will not be precise. So better to have absorption less than 0.1. Then you'll get a curve. It looks somehow like this. The higher concentration you have, the higher absorption you will get. But we don't need this. We need the linear curve. To get the linear curve, we need to get the equation of the curve. Now we have the equation. The y is the absorption and the x is the concentration. Let's say I have a protein sample of an unknown concentration. I'll take 1 microliter of this solution, mix it with Comasi brilliant blue, measure the absorption, and then calculate the x, and then I will get the concentration of my proteins. To discuss the ELISA, we have Alex and Laarni.
Moving on to the next part, let's take a look at ELISA. First, what is ELISA? ELISA is a test method that has a range of advantages. It provides for quick and specific quantitative assessment while being efficient and simple to use. An enzyme-linked immunosorbent assay, also called ELISA or EIA, is a test that detects and measures antibodies in your blood. This test can be used to determine if you have antibodies related to certain infectious conditions. Antibodies are proteins that your body produces in response to harmful substances called antigens. The most commonly used enzyme labels are horseradish peroxidase and alkaline phosphatase. Other enzymes have been used as well. A large selection of substrate is available commercially for performing ELISA with an HRP or AP conjugate. However, certain equipment is necessary before a laboratory may begin working with ELISA. Let's take a look at the materials needed for ELISA. Depending on the test system used, various laboratory devices may be needed for ELISA processing. Some test kits require further devices, such as an incubator for constant temperature during test implementation. Let's start by looking at pipettes. It is a common piece of lab equipment. Pipettes, of which there are several types, are used in ELISA testing to transport a precisely specified volume of a solution to the walls of a microtiter plate. Using a pipette typically yields improved consistency by reducing pipetting errors and decreasing variability in incubation time from the beginning to the end of plate. It also reduces repetitive motion. Next one is microtiter plates. Microtiter plates are cleaned using the ELISA washer. After each incubation stage, the ELISA plate must be washed with washing buffer, excluding the incubation with the substrate. It is appropriate for cell-based and biochemical studies intended to detect and measure protein species. Numerous immunodiagnostic and DNA-based diagnostic test kits use ELISA products. It permits the use of the standard laboratory tests that can be applied to numerous samples at once. The common test for assessing a protein mixture and assisting in establishing the presence of the target protein is the ELISA test. The last material needed is the ELISA reader. The ELISA reader is also quite important. This is a spectrophotometer that can measure optical density and determine sample concentration. It plays an essential role. The high cost as well as the space and maintenance requirement is often an obstacle to starting with ELISA analysis, especially for small laboratories. The instrument is designed to fit standard 96 well plates and will provide quantitative information about the extent of the response for each individual well of the plate.
Methods of ELISA. ELISA is one of the simplest blood tests to do. It is fast and only takes a blood sample from the patient. The first step is the antibody is linked to a polystyrene plate, which has a solid surface, and it is attracted to bacteria, other antibodies, and hormones. The next one is the antigen-antibody combination is placed in an antigen-coated microtiter and free antibodies are eliminated by washing. After that, a second antibody that is specific to the first antibody is frequently conjugated or linked with an enzyme. Washing the plate removes any free enzyme-linked secondary antibodies. And finally, the substrate is added. This enzyme converts the substrate to a colored product that may be detected by spectrophotometry. ELISA detects HCG protein, which indicates pregnancy. A combination of blood or urine sample and pure HCG linked to an enzyme is given to the system. If HCG is not present in the test sample, just the related enzyme binds the solid surface. The more the substance of interest is present, the more reaction occurs, and the less linked enzyme binds to the solid surface. These reactions are typically recognized by a change in the color of the solution.
Now let's take a look at the three types of ELISA. The first one is indirect ELISA. Antigen is coated to the microtiter well. Since many primary antibodies can be used with a particular secondary antibody that has been labeled, indirect ELISA offers more versatility. One of its drawbacks is the potential for secondary antibodies to cross-react with the immobilized antigen, which could enhance background noise. Second one is the sandwich ELISA. It entails coating with the microtiter well with an antibody. It is a method that scientists use to check whether an antigen or an antibody is present in the sample. The capture antibody and the detection antibody with each detect a separate epitope of the antigen are coupled together with the analyte to be analyzed. As a result of its sensitivity, this format is widely utilized. Lastly is the competitive ELISA. The antigen-antibody combination is put into an antigen-coated microtiter well. It uses an inhibition antigen. By identifying interference in an anticipated signal output, it is used to determine the quantity of an antigen or antibody in a sample. In competitive immunoassays, the analyte is labeled, while in endometric immunoassays, the reagent is labeled.
What is salivary amylase? Salivary amylase is an enzyme found in the saliva. It is in charge of converting carbohydrates into simpler sugars. When we eat, salivary amylase is generated by the salivary glands and released into the mouth. Saliva is the most abundant and easily accessible source of amylase. It then converts the carbohydrates in our food into simpler sugars. It influences oral starch perception, metabolic signaling, and plasma glucose responses to ingested starch. This salivary enzyme is responsible for converting complex carbohydrates like starch into simpler sugars like maltose. An enzyme's primary function is to accelerate a reaction. To produce products, most enzymes work with only one reactant, known as a substrate. The most remarkable feature is that enzymes can be regulated from low to high activity and vice versa. Changes in pH, temperature, concentration, and presence of inhibitors have a significant impact on enzyme activity. Each enzyme works best at a specific pH and temperature, with the denaturation decreasing activity at values above and below that point. pH of the incubation. Any enzyme has a maximum activity pH. The interaction of the substrate with reactive groups provided by the side chains of amino acids at the binding site occurs during substrate binding to the active site of an enzyme. The pH of the incubation medium may influence the ionization of both the substrate and the amino acid side chains, affecting binding. It may also alter the ionization of reactive groups that catalyze the reaction, albeit this is less likely in the microenvironment of the catalytic site when it's occupied by the substrate. Extreme pH levels may also damage the enzyme's tertiary structure, distorting the active site or even denature the enzyme protein. Different proteins will react differently to severe pH levels. The temperature of incubation. Temperature affects the rate of an enzyme catalyzed reaction due to two opposing variables. As with any chemical reaction, the rate rises with increasing temperature because the activation energy of the reaction is more easily delivered at higher temperatures. This indicates that the creation of product increases dramatically between 5 and 50 degrees Celsius. Heat denatures enzymes since they are proteins. As a result, at higher temperatures above around 55 degrees Celsius, the protein loses functions rapidly due to irreversible denaturation. Concentration of substrate. The rate of starch breakdown will be slower the lower the amylase content. The breakdown of starch will occur more quickly if the number of amylase is doubled since there will be twice as many active sites. Long protein molecules that have been folded into an amorphous globular shape make up enzymes, as long as there is substrate available for the enzyme to bind to. Increasing enzyme concentration will accelerate the reaction. The reaction won't accelerate after all of the substrate has been incorporated since there won't be anything left for further enzymes to bind to. Enzyme activity rises when enzyme concentration rises because there are more enzymes to form complexes with substrates. Presence of inhibitors. Inhibitors are compounds that slow or stop the activity of enzymes. Competitive inhibitors are molecules that are chemically similar to the substrate and can bind to the enzyme but not react with it. They start competing with the substrate for the enzyme's active site. Amylase inhibitors inhibit salivary and pancreatic amylase activity in vitro and in vivo. When consumed in large quantities, they can impair mammalian growth and metabolism. Non-competitive inhibitors are molecules that differ from the substrate and thus do not bind to the active site.
This is the end of our presentation. Thank you.